Hello everyone, welcome back to our plant physiology note series part 3. In this video, we'll cover cytokinins hormone. And in cytokinins, we'll see discovery, chemical nature or basic structure, bioassay, physiological role and mode of action. So let's start the topic. The discovery. The cytokinin was discovered by Miller et al. In 1950 while working in Skook laboratory at University of Wisconsin on the growth of tobacco pith callus in culture. Initially, they attempt to promote callus growth by adding various substances like vitamins to culture medium, but none produce positive result. During this time, they notice an old bottle of DNA that have been stored in the laboratory for several years. Out of curiosity, they added the contents of the DNA bottle to the culture medium and were surprised to observe that the tobacco pith callus continued to grow actively for a longer period. Further investigation revealed that the active substance in the aged DNA was 6-furfurylaminopurin, which they isolated and identified. Because of its special effect on promoting cell division, cytokinesis, the compound was named cytokinin and it is also known as kinetin. Some naturally occurring cytokinins include coconut milk factor and zeatin. The chemical nature of cytokinin are adenine derivative with a side chain at the N6 position of the purine ring. The important cytokinins are number 1 kinetin. This is the structure of kinetin. And number 2 zeatin, naturally occurring in meat. This is the structure of zeatin. Now we'll see the bioassay of cytokinins. The most common test, tobacco callus assay or root meristem assay. The principle of this bioassay is cytokinin stimulates cell division in cytokinin deficient plant tissue or callus culture. The degree of cell proliferation or growth of the lateral bud is proportional to the cytokinin concentration. The procedure of the bioassay: excise tobacco leaf segment and place them on cytokinin free medium. And number 2, observe the formation of callus undifferentiated cell mass. And number 3, treat the experimental Kelly with different cytokinin concentration. After 7 and 14 days, measure the increase in fresh weight or number of dividing cell. And we'll see the more cytokinin, the greater the cell division and callus growth. These include the more concentration of cytokinin and more cell growth occur. Now, our next topic, physiological effect or roles of cytokinin. First, promotion of cell division. The cytokinin stimulates mitotic activity in roots, shoot, callus tissue, and it works synergistically with oxygen to determine organogenesis, means whether it's shoot or roots. Number 2, breaking apical dominance. Cytokinin promote lateral bud growth by counteracting oxygen, and cytokinin also used in horticulture to encourage branching. Number 3, delay senescence. Cytokinin maintains chlorophyll content, protein synthesis and photosynthetic activity. Cytokinin also prevent leaf yellowing. Number 4, shoot formation in tissue culture. When cytokinin combine with oxygen, cytokinin determine shoot or root differentiation. When high concentration of cytokinin with oxygen ratio, the shoot formation occur. When low concentration of cytokinin and with oxygen ratio, the root formation occur. Number 5, vascular differentiation. It stimulates formation of phloem elements and work with oxygen to regulate xylem and phloem development. Number 6, nutrient mobilization. Enhance mobilization of nutrient to actively growing tissue. And number 7, seed germination and growth. Cytokinin can stimulate cotyledons expansion and delay senescence in developing seed. Now our last part, mode of action of cytokinin. Number 1, cytokinin perception. Cytokinin detected by histidine kinase receptors on the plasma membrane, and it triggers a phosphoryl signaling cascade inside the cell. Number 2. Activation of gene expression. Cytokinin induce the synthesis of specific messenger RNAs, mRNAs for protein that promotes cell cycle progression. In these gene activations, the key genes are involved cyclins and cyclin-dependent kinase (CDKs). Number 3. Cell cycle regulation. Cytokinin stimulate transition from G1 to S phase in the cell cycle, enhance cell division. Number 4. Interaction with auxin. Together, auxin and cytokinin determine pattern of organ development, means root, shoot, callus differentiation. So this is the cytokinin, and you can download the PDF file from description. And thank you. Bye bye.